Hi everyone, welcome to this set of notes. This is Mitosis. We're in Chapter 8. The standards that go along with mitosis, we're still working on 1.6, which is to determine the relationship between cell growth and cell reproduction. You've already done the notes on the cell cycle, so you've talked about when the cell grows, when the copy of the DNA is made, and it's two separate growth phases. So now we're going to talk about the reproduction part of the cell cycle, and you need to be able to identify the stages of mitosis given a visual image, and the best way to do that is to know the locations of those chromosomes because they're the most visual part of the stages. You also need to be able to demonstrate the movement of chromosomes during mitosis, so describe how those chromosomes move. So think about this. How long do you think it takes for mitosis from start to finish? Hopefully you said that it varies, because it does. Mitosis can happen as quickly as about 20 minutes, and it can take as long as up to 24 hours. It depends on the cell and the requirements for that particular type of cell. These are the steps of mitosis: Prophase, Metaphase, Anaphase, and Telophase. Telophase and Cytokinesis are often grouped together. However, sometimes you may see Cytokinesis as a separate part. I would recommend the easiest thing to do is to remember PMAT, the first letters of each one. For me as your teacher, the PMAT would stand for Please Make A's on tests. But if you can come up with some sort of acronym that will help you remember the order in which these occur. So Prophase, Metaphase, Anaphase, and Telophase. Here's a basic diagram of what those stages look like. In Prophase, you have those X-shaped chromosomes. Remember that X-shaped chromosomes mean that the cell is getting ready to divide. So you see those in Prophase, and they're just kind of hanging out. The nucleus here is somewhat visible. They usually do it as a dotted line or a dashed line. Metaphase, the nucleus is completely gone, and you see those X's lined up, those chromosomes lined up in the center of the cell. In Anaphase, you have torn those sister chromatids apart, so the chromosomes are no longer X-shaped. They are in fact these right here. And we'll demonstrate in class why they take on this shape, kind of like a V. But if you imagine a piece of string laying on your desk, if you put your finger in the middle and pull the middle of the string towards you, the string will look like a V, and that's in fact what's happening here. So there are the chromosomes at the end of those fibers. And then in telophase and cytokinesis, you have the chromosomes looking like a rod shape. By the time the cell enters interphase again at the end of mitosis, the nucleus is a large black dot again, because the rod-shaped chromosome has unraveled and become chromatin. So now that the overview is done, let's get down to business. Before mitosis can occur, there are several things that happen in the cell. And one is the centrosomes, the centrioles, and the mitotic spindle. Centrosomes, which you can see here, and this, if you imagine like a golf ball with lots of little holes in it. So you have the centrosome. Inside there, you have these centrioles. And the centrioles are microtubules, so kind of like cytoskeleton type structures that grow from here. So the centrioles are kind of like a base, and you're going to build off of it in a particular direction. So in our electron micrograph picture that they have highlighted with different colors, the white circles, there's two, there's one on each end of the cell. These are your centrosomes, so the white circle represents your centrosome, one on each side of the cell. In there, you see these little T-shaped structures, the little black ones, those are your centrioles. Now your centrioles are responsible for growing and spitting out all of these little microtubules. The microtubules you can think of as cables. These are really strong, somewhat rigid strings. And these somewhat rigid strings start to grow out in all directions, and those are represented in green here. So they grow out from both sides. As these microtubules grow towards the center of the cell, the blue represents where you would find the chromosomes, and the red represents the centrosome of those chromosomes. So as the microtubules grow, they randomly shoot out and they hopefully connect with the centrosome of our X-shaped chromosome. And all of this green is the mitotic spindle. So that's the name for all of this mess, because it's like a mass of microtubules growing out of the centrosome. And as they grow towards the chromosomes, they attach to the centrosomes. We're going to talk a little bit more about that as we go through mitosis, but I am going to reference the centrioles and the mitotic spindle. So that's what I'm talking about there. Step 1 is prophase. And there are things that happen in prophase. First of all, it's important to remember that during the cell cycle, during S phase, before we ever start mitosis, during S phase, the DNA was copied. So there's lots of chromatin in the cell. In fact, there are 2 sets of chromatin. And this is the point, the first phase of mitosis, where the chromosomes condense and start to form those X-shaped structures. In prophase, the nuclear membrane starts to break down as well. If we're going to pull those chromosomes to opposite sides of the cell, then we don't need the nucleus there getting in the way. So the nuclear membrane breaks down. The centrosomes appear at this point. So here they are. You see that characteristic T shape again. So one going up and down and one going left to right. And you see 2 of them. They appear and they start to migrate towards opposite ends of the cell. And the mitotic spindle begins to form. And the mitotic spindle in this drawing is represented by the lines between the centrosomes. Quickly pause the video and see if you can recap the things that happen in prophase. Next we have metaphase. And in metaphase you can see we still have our centrosomes with the 2 centrioles inside. You can see all of the fibers, so you can still see the mitotic spindle coming out of them. But what is happening now is that the spindle fibers, as they are launched out and they grow, by chance they happen to hit the centromere of each chromosome. And since you have 2, one on each side of the cell, the centrosome will end up with 2 spindle fibers attached to it, and they both start to retract and pull, and the chromosomes end up lining up in the middle of the cell. Kind of like this dramatic tug of war between this centrosome and this centrosome. They've attached their fibers to the middle of the chromosomes, and now they're both pulling on them. And that pulling action lines up the chromosomes in the center of the cell. And this is metaphase. One way to help you remember this is metaphase, middle. Chromosomes are in the middle of the cell during metaphase, and they are still X-shaped. Take a minute and see if you can recap what was going on. Now on to anaphase. In anaphase, the chromosomes that were formerly lined up in the middle have now been pulled apart. So in this epic tug of war, the sister chromatids actually separate so that one of the sister chromatids goes to the left and one goes to the right. This ensures that each cell has a complete copy of the chromosomes. you can pause the video and write down this information, but I'm going to move on. Again, pause the video, see if you can describe what happens in anaphase. Telophase and cytokinesis. I'm going to group these together. However, like I said in the beginning of the video, some texts and some videos may separate these into two separate steps. It's also important to realize that it is different in animal cells and plant cells. So let's talk about animal cells first. In telophase, the nuclear envelope begins to reform. So the chromosomes are now on opposite sides of the cell, and the nuclear membrane starts to kind of pull itself back together. The chromosomes start to unwind into chromatin, and the cleavage furrow, this contractile ring, appears, and it starts to tighten, and you get this cleavage furrow, is what this is called. This continues to contract until it separates the two cells. The yellow things are our centrosomes, and you can see that the centrosomes are still there. The nucleus has reformed, and inside there they've shaded it all blue because our rod-shaped chromosomes have started to unwind into chromatin, which makes them a solid shape under the microscope. And in the end, you have two new cells. So let's talk about plant cells. Plant cells work a little bit differently. Again, the chromosomes are at opposite ends of the cell, and just like in animal cells, the nuclear envelope begins to reform, and you can see in the second picture that that nucleus is there. Just like in animal cells, the chromosomes begin to unwind. In the last step, instead of having that cleavage furrow and pinching off in the middle, the Golgi apparatus actually starts to build what's called a cell plate. So the Golgi apparatus builds these vesicles, and the vesicles start to fuse, and the plate forms, and you have two entirely new plant cells. So pause the video and review the differences between the plant and animal telophase. All right, this is the exit ticket that we would have done in class, but you do need to come up with a memorization tool for the steps in mitosis. You have a quiz on Monday, and on Monday bring your foldable and any printed pictures that you'd like to glue in, and we will review this again, but briefly, in class.